Greetings, everyone, and welcome to today's webinar, Educational Webinar, Implementation of a Preemptive Pharmacogenomics Program at Pediatric Health System. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image and follow the process to obtain your credits. And with that, I'd like to now welcome our speakers, Dr. Stephen J. Melnick, Chief Department of Pathology and Clinical Laboratories, Nicklaus Children's Hospital, and Dr. David B. Mancuso, Executive Director of Pharmacy and Laboratory Services. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Welcome, Dr. Melnick and Dr. Mancuso. You may now begin your presentation.
Thank you, and welcome everyone. First, we have nothing to disclose on our end. Below is a disclaimer that's provided by Thermo Fisher Scientific. Brief word about our hospital. Nicklaus Children's Hospital is actually 75 years old. It started as Variety Children's Hospital. It became Miami Children's Hospital in 1983 and then Nicklaus Children's Hospital in 2014. It's a 307-bed hospital. It is South Florida's only licensed specialty hospital exclusively for children, with approximately 800 attending physicians and more than 500 pediatric specialists. The learning objectives for the session today: one is to understand the clinical impact of pharmacogenetics. We will describe some of the challenges to clinical implementation of a pharmacogenetics program, to recognize major resources for clinical guidelines for the implementation of the program, and to describe key features for program development. I'm going to pass the next conversation to Dr. Mancuso, who's going to provide you with an overview of pharmacogenetics.
Hello and welcome everyone. My name is David Mancuso and I am the Executive Director of Pharmacy and Laboratory Services here at Nicklaus Children's Hospital in Miami, Florida. We are very excited to be here today and being able to share some of the practical knowledge and experiences that we have related to the implementation of pharmacogenomic testing into clinical practice. Before diving into the topic of clinical implementation, we would like to talk about what pharmacogenomics is and how it affects patient care. Pharmacogenomics is the branch of pharmacology that uses genetic information in order to understand variability and drug response. And by understanding this variability, we can choose medications or medication doses for our patients more effectively. As you see in the slide, about 97 to 98% of our population have at least one genetic variant that affect how they respond to medications. Therefore, this information is very relevant since it affects almost every patient that we encounter. On the right-hand side, we can see the difference between traditional treatment and the precision medicine approach that offers pharmacogenetic testing. And that picture pretty much illustrates what we often see in clinical practice. When you prescribe a medication, the majority of the patients will react as expected, but there is a subset of patients that will have unexpected adverse drug reactions, and another percentage of patients will experience a therapeutic failure meaning that they will not react to the treatment at all. Pharmacogenetic testing is used to identify that subset of patients and help us to increase the likelihood of a more effective response to treatment and help us to reduce the chance for adverse drug reactions. In summary, pharmacogenetic testing, when it's done the right way, it has the potential to change the entire way we prescribe medications and the way we take care of our patients. Now, I would like to show you in the next slide some statistics that illustrate the impact of PGX and population health. The data that you see on the slide was extracted from the website that you see on the slide as well, and it shows that the top 50 prescriptions that are being dispensed in the United States are displayed on the slide. Just by looking at these medications, we find out that 32% of those medications had published pharmacogenetic clinical guidelines. They are associated with pharmacogenetic guidelines. And this is from 2021. And the top 50 medications resulted in 500 million prescriptions that affected 125 million patients. A similar scenario can be seen for the top 100 medications that are being prescribed in the United States. That translates to about 1/3 of the population in the United States affected by a pharmacogenomic interaction, showing that the contribution of pharmacogenetics to population health is very significant. So how about pediatrics? Is the impact on pediatric populations significant? So on December of 2020, we collaborated with other institutions on a publication that highlighted that prescribing of medications with the potential for pharmacogenetic interventions was not only a possibility, but was very common and prevalent in pediatric patients. Fast forward to 2024, and we found that another publication looking into Medicare recipients clearly found and established that there are opportunities to implement pharmacogenetic guiding treatment in this population. So we can see that the conclusion that is drawn year after year is that pharmacogenetic testing should be considered in the pediatric population as well. Because we talk about pharmacogenetic testing being able to help to minimize adverse drug reactions and therapeutic failures, we would like to give you in the next slide an idea of what is the impact of non-optimized therapies in the United States. Unfortunately, as you see in this picture, this is a sad reality, but across various therapeutic classes, there is a significant percentage of patients that experience a suboptimal response to standard treatment. That accounts for about 50% of patients failing treatment. The consequences of this is not just clinical, but it is also a financial impact. As you can see on the right-hand side, adverse drug reactions are responsible for more than 100,000 deaths among hospitalized patients annually, and is placing a $2.4 billion burden in our healthcare system. When we look at non-optimized therapies, the cost is staggering as well. A monumental $528 billion are spent annually trying to solve this issue. We often get the question of how much it costs to implement pharmacogenomic testing, but when you look at these numbers, the question is no longer about whether we can afford to implement pharmacogenetics, it's whether we can afford not to implement it. With that, I will pass it to Dr. Steven Melnick that will talk about some of the existing challenges into clinical implementation of pharmacogenetic testing.
Thank you. So I'd like to describe some of the basic areas where implementation of a pharmacogenetics program can have some challenges. The first point is to establish the need. And as Dr. Mancuso clearly illustrated, there's a great need for having pharmacogenetics as part of the continuum of care for patients. However, more basically, it is very clear also that there are hundreds of actionable genetic variants with dosing guidelines for FDA-approved drugs. And that represents a significant percentage of FDA-approved drugs and of prescriptions as well. And this number is continuing to grow. So clearly the need is there. However, what about the evidence? Well, over the last 10 years and further, there's a growing body of evidence from clinical trials of genotype-guided dosing. Much of this information has been evaluated, curated, and annotated by different organizations that provide standardization for this process, and we will describe those. There are also a lot of studies that point to the cost effectiveness of implementation of pharmacogenetics. So certainly the evidence is there as well. But there's a laboratory testing component. There are a number of decisions that have to be considered, whether to focus on microarray testing or sequencing. Is the panel sufficiently comprehensive? Should they be tailored or limited? What about the expense and the turnaround times? Are the turnaround times going to be sufficient for clinical actionability? These are important considerations that we will talk about. As I mentioned, there's also the translation of the genomic data into clinical actionability and dosing guidelines. And there's also a great need for standardization of each step in the process. These organizations, which we will delve into more, provide that level of standardization. So there's a lot less for us to have to do. They're doing the heavy lifting. Informatics is a vital component to this process. Without an informatics component, it would be virtually impossible to have a successful pharmacogenetics program. This involves taking enormous amounts of data and placing it through sophisticated algorithms in order to assimilate the information and provide drug gene information and clinical dosing recommendations. It's necessary to sift through hundreds of different variants related to dozens of different genes in this process, and also account for various ethnic variations in drug response. So clearly, there's an important and vital need for informatics. And finally, decision support. This is really the end of the process, and it needs to be done in such a way, in our opinion, so that physicians can have access to the information in a seamless and time-relevant manner. We will describe that as well. Very simply, this is a multi-step process, starting with the genotype of an individual, which gets converted into a haplotype, diplotype, then phenotype, and then the therapeutic recommendations. The first two steps are performed in the laboratory. Typically, we will generate a VCF file or other compatible format, which is fed into the translational software and then into the informatics system to develop the clinical guidelines and then the clinical decision support for physicians. But at each step of the process, standardization is vital. I can't stress that enough. But fortunately, as I mentioned, a great deal of that work has been done for us by these different consensus organizations. I will just reference them briefly because Dr. Mancuso will be elaborating on them later in the discussion. The Association for Molecular Pathology has for at least 10 years been able to define most appropriate genes to focus on for pharmacogenetic testing and provides level of evidence for different variants that should be included as a minimum in different panels. Pharm.GKB is a pharmacogenetics knowledge base, which is very vital. They basically will go through and access all the information that is available from clinical research and then curate it and annotate it and develop it and prepare it for clinical relevance. Pharm.var is a repository for the annotation or nomenclature that's used in pharmacogenetics, and that is very standardized, and pretty much everyone is utilizing that particular nomenclature, particularly the star allele nomenclature. CPIC is the clinical pharmacogenetics Implementation Consortium. It's a global consortium which takes all of that information and basically presents clinical guidelines and dosing recommendations. And PharmCAD is a bioinformatics tool which is available for everyone to use. And it's a very useful tool where you can take a BCF file for a patient that has pharmacogenetic testing. That information can be processed and generating a report. It also will have disclaimers when insufficient numbers of genes or alleles are interrogated so that nothing will be missed. And I will talk about the implementation of our program now. Dr. Mancuso and I, about 5 years ago, began the journey into the clinical implementation, but it rests on a foundation of work that was done by a colleague of ours, Dr. Jennifer McCafferty, who explored the viability of pharmacogenetics before we even got to this point. She was at that time director of a research institute and conducted some vital research in that area with some publications, paving the way for us to implement a clinical pharmacogenetics program, which, as I mentioned, started about 5 years ago. So conceptually, these are the things that we start with. Obviously, there has to be some leadership in defining the program, and there are specific goals that have to be considered. Initially, early on, we talked about whether we were going to have a preemptive program, meaning that the testing would be performed before patients began their journey in the health system and had any medications prescribed, or a focused or reactive methodology where patients who might be in a particular treatment protocol in psychiatry or oncology, for example, have specific drugs that they're likely to use. We would focus, though, the pharmacogenetics based on that. There's also the establishment of a team. Obviously, pharmacy and lab are important, but IT is vitally important because nothing could be accomplished in terms of the integration of the data that we produce without involvement of our colleagues in the IT department. And importantly, also our physician champions. We have very broad discussions with our clinical colleagues because they have to understand our vision and what the potential applications for pharmacogenetics in each of their subspecialties will be. There's defining the test methodology that is either going to be comprehensive, which is the path that we've taken because we want to focus on a preemptive program. So therefore, not knowing what drugs a patient may be receiving, we have to be as broad as possible in terms of the evaluation of their genomic profile. And then there's also the possibility of a focus or more limited panel. EMR integration is vital. In the laboratory sense, we integrate with the EMR all the time, but we're not simply producing one result or a small number of results that get interfaced. This information is going to be interrogated through an informatics resource and then directly input into the EMR. So there's another stage there that's vital and requires a great deal of effort on the part of our IT department. There's decision support, which is also a very vital part of the process, without which we don't believe that a pharmacogenetics program can really be viable. That means having the information produced, having it processed through an informatics system, and then displayed for physicians in a seamless manner and a very timely manner. So this just outlines the various interfaces that are involved with our PGX program and our partners in other parts of the institution. So at Nicklaus Children's Hospital, this is what we did. So we started the journey as a collaboration between pharmacy and laboratory about 5 years ago. We decided very early on we were going to work toward developing a preemptive pharmacogenetics program. In the first phase, we were going to roll it out in these particular areas, mainly because physicians who work in these areas already had experience with pharmacogenetics. They are an important part of their current practice, so that became the best opportunity. Those were our physician champions. But we also recognize that even though specific medications are used in each specialty, that within each specialty, other medications are used that cross other boundaries. So therefore, it became clear that it was important to have a broader panel to make sure that the needs of all the patients are taken into consideration, regardless of which subspecialty their primary care is provided. Next, we will develop a more system-wide approach, but that will follow in this year and in the subsequent years. We developed in-house pharmacogenetic testing, and I will discuss that in a little bit more detail. We had to make a decision about the informatics. We don't have the capability within our institution to develop a robust informatics program and knowledge base, so therefore we sought support outside the institution with an informatics vendor, and I will discuss with you some of the things that we look for when making that decision. Importantly, we have pharmacogenetic counselors to support our medical staff. We have a subcommittee that reports to the Pharmacy and Therapeutics Committee related to pharmacogenetics. Also very important at an institutional level, as I mentioned, engaging our stakeholders in different areas is vital, but so too is engaging administration. Ultimately, administration will support the funding of this initiative, and so it's very important to have a transparent and robust conversation with various members of the hospital administration. As I mentioned, IT is vital as well, and we've had those conversations before we went any further. Laboratory and pharmacy, of course. And then the discussion about preemptive versus reactive pharmacogenetics. We opted for a preemptive approach, but here's the basic difference. From the point of view of ordering, if you have a reactive panel focused on a particular application, it's relatively small. It will be ordered at the time the patient is diagnosed, perhaps. Maybe the patient will be on empirical therapy for a short amount of time until the results of the testing becomes available, and then that information will be used to either continue or modify any subsequent therapy. The preemptive approach basically means before the patient is offered any therapy, the information is already in the system. Turnaround times for reactive testing can vary. The panel that we are using, even though it's preemptive, is about 7 to 10 days. It's a complex process which involves about 4 days, and I'll describe that. But if you're going to use a vendor outside your institution, it will generally take at least 14 to 21 days to get results. So it's not really very timely, and it depends on how you wish to use that information. The preemptive testing means basically the information is already in your system at the time the patient requires any medical therapy. So there really is no turnaround time. From the end user perspective, it's unlikely that if a small panel is used or is used infrequently, the hospital is going to invest in the necessary integration of this level of testing into the EMR. So much of the responsibility will fall on the clinician to take the data that's obtained in a report and do their own evaluation for the gene-drug interactions for their particular patient. It can become very time-consuming, and not many physicians are likely to engage that process. However, with preemptive testing, we have the opportunity to have a full-fledged integration of decision support from the informatics and into the EMR so that physicians can receive alerts, they can query different drugs and get immediate feedback without having to do much of anything else other than care for their patients, which is their primary obligation. The financial considerations are important. Reactive testing costs can be substantial. If using reference laboratories in-house, it could be a little bit less expensive. The costs are similar for preemptive testing. However, we have a different value proposition. We believe, and we've had this conversation with our hospital administration, that really what pharmacogenetics brings to the table is not a diagnostic test, but is an opportunity for ensuring quality and safety for medication use for our patients. Every hospital has some obligation and some body which focuses on quality and safety within the organization. Hospitals spend enormous amounts of money for quality and safety without focusing on reimbursement. We believe that pharmacogenetics, as really clearly established by what Dr. Mancuso was sharing with you, provides enormous opportunities to establish that level of quality and safety for patient care, reducing adverse drug reactions, and ensuring optimal therapy for patients. So it's a totally different calculation. Test methodology, reactive testing focuses on a small number of genes and small number of variants, where we are utilizing an array-based methodology which queries multiple genes and hundreds of variants. So the next consideration is the choice of the assay that's being used. Well, some considerations that are important are whether the laboratory has the capability to do it already, or if they need to develop these capabilities from scratch. What methodology should be used, microarray, NGS, RT-PCR? What are the financial implications? How is this going to be integrated into the existing laboratory process? And what is the clinical relevant time to result? And then also integration with decision support platforms. So these are some basic considerations. At Nicklaus Children's Hospital, we started with the fact that we had an existing molecular pathology and precision medicine laboratory. So basically, we had all the ancillary equipment that was needed. We did obtain a microarray system and a robotic system for handling specimens, reagents, which is very vital, but everything else existed in the laboratory that we might need. We went through the analytic validation process, which took about 6 weeks. We then honed in on our data management and analysis with the software that was provided and worked through that process. We then worked with an outside vendor, which I'll discuss a little bit more detail, which managed the information for us. And this is an outline of the process. So we start with, excuse me, an order that's placed in our system, which is Oracle Cerner. Once the order is placed, the sample is obtained. It could be a buccal swab or blood. It is sent to the laboratory, accessioned, genotyped in the laboratory. Once the data has been analyzed, a data file is prepared and submitted to a location in the cloud environment. Our colleagues at ACTX will retrieve the information that we provide in either a BCF or text format. They will process the information and then it's reviewed on our end once it is completed before it's released into the EMR. Once everything is okay and our pharmacists who review the information find it acceptable, it will then get released, and then we will describe to you what that looks like for our providers and our families. It's really very seamless and very easy without much effort, but you'll see how that unfolds shortly. So in the laboratory, just briefly, the workflow is about 4 days. We start on day 1 with the extraction and then the amplification of the DNA. There are 2 components to that. Most of it involves an isothermal reaction. However, some areas in the genome need to be further resolved, such as CYP2D6. We need more resolution. Therefore, we use different primer pairs to really amplify further some of the targets that are important to us. Then all of that gets recombined. The next day, we do the fragmentation of DNA, overnight precipitation. The next day is preparation for hybridization after resuspension, then the hybridization in our microarray system. And on day 4, there's the ligase wash and imaging, and then we analyze the data on the following day. So as I mentioned, we didn't have the capability to do the clinical decision support with an informatics in our own institution. So we went and sought support outside the institution with an informatics vendor. The things that we were looking for was the experience of the vendor and longevity in the marketplace. We wanted to make sure that that particular vendor was agnostic to EMR, meaning they could work with Cerner, they could work with Epic, they could work with Allscripts, they could work with a host of other EMRs. The problem, we didn't want to be faced with a problem like we were the first one with Cerner that they ever engaged. That would not work very well, and it would add a lot of time to the process. Other thing is CLIA certification, which is very important because you're handling all of this information. We want to make sure that the level of attention that they pay to the process is also evaluated by other regulatory agencies. There's the data curation knowledge base that they use. We want to make sure that they're using primarily CPIC guidelines, but if they're using anything else, we want to make sure we understand what the level of evidence is that they're using to add their content to the information that we provide. We also want to make sure that we understand how the data will be accessible for both patients and families, for physicians and families. EMR support is important, as I mentioned, so the informatics provider needs to work with our EMR, and that requires a lot of work with our IT department. Together, they will build the alerts, display the data, and give physicians access to the supporting information and the seamless integration for immediate decision support. Just very briefly, it's important when you start on this program to develop a timeline. You want to have that as well as milestones. You will notice that this occurred during the COVID pandemic, so we were delayed in some areas, so it didn't quite happen exactly as we had wanted, but obviously we all understand why that occurred. But the important point here is to define each of the steps in the process that require some effort by different members in the organization, give them the appropriate amount of time to do that. A lot of times there will be overlapping of different activities, but you want to map it out and adhere to it as much as you can and develop milestones, acknowledging it may not always work out as well, but this helps keep everyone on task. And this is really the important point of developing such a timeline. And finally, the importance of the process internally. When we use information that we obtain from our informatics provider, we want to make sure that they stick to certain guidelines, but also if they have other content that they want to add, we need to evaluate it. So we need a process in which to do that. So that starts with an internal review on our own end, passing it through the pharmacogenetics subcommittee to review it, then pass it on to the pharmacy and therapeutics committee, then implement those changes and educate prescribers, nurses, and pharmacists. And I will now pass on the conversation to Dr. Mancuso, who will talk about the clinical guidelines.
And thank you, Dr. Melnick, for such a comprehensive explanation of the process and outlining some of those challenges into clinical implementation. And right now, I would like to shift our attention to the available resources for implementation of pharmacogenomic testing into clinical practice. When we were looking to implement PGX into clinical settings, we quickly realized how important it is to rely on evidence-based guidelines that are robust and well curated. We are fortunate to have several key resources to our disposal, and some of them were previously mentioned by Dr. Melnick, and I would like to go over in more detail, but before I continue, I want to make sure that everyone understands that these resources are essential to clinical implementation. These resources are extremely important, and the majority of them are not optional. You need to use these resources. First, we have CPIC, or the Clinical Pharmacogenetics Implementation Consortium. As many of you know already, these consortium guidelines are the gold standard, and they help to interpret genetic test results and applying them to medication management in a practical way. A point that I would like to make, which is important, is to understand that CPIC won't tell you when to test, but rather they assume that the genetic information, the PGX information, is already available. Next is Pharm.D. This is the Pharmacogene Variation Consortium. It's another great resource and is the home for the pharmacogenomic nomenclature, and is serving as a centralized data repository, and it houses the information for gene variants that have clinical relevance. And there is also Pharm.GKB, which stands for the Pharmacogenomic Knowledge Base. This resource provides comprehensive information that contains gene-drug interactions and their impact on drug response. Across the continent in Europe, we have the Dutch Pharmacogenetic Working Group, and this is similar to CPIC, and they offer guidelines that are highly regarded and they're often adopted internationally. And lastly, we have the Association for Molecular Pathology that provides recommendations for genotyping and allele selections, and they set the standards for proper laboratory testing. It is important to note that these guidelines are available for any healthcare provider, and they are free and accessible online. Now, moving to the next slide, I wanted to show you the evolution of these guidelines and to illustrate that pharmacogenetics is not a static science, but rather is a very dynamic field, and it continues to evolve. There is a need for a commitment to staying informed and updated with these changes and these guidelines. And if you're trying to implement pharmacogenetic testing into clinical care, we will strongly recommend to adhere to these guidelines and to use these clinical resources that we mentioned throughout the presentation into your adoption to pharmacogenomics. And as you can see on the slide, and from 2011, the clear evolution of these guidelines. And the next slide, you will see also we have nowadays 26 guidelines, 27 genes, and more than 100 drugs that are ready for implementation. And on that note, I will pass it on to Dr. Melnick that will show you what type of genetic panel are we using in our institution.
Thank you. So I'll share with you the panel that we are using. First, on the left column, these are labeled as CPIC genes. CPIC has qualified a number of different genes and variants that can be used. Most of them are reflected here. There are numbers that we are not using in particular. Much of this is guided by the collaboration that we have with our informatics vendor that has content for some genes and gene variants, but perhaps not others. So basically, we reconcile the testing that we're doing, the availability of different genes and variants, and align that with the content that is available by our vendor. The next two columns show the genes for which our software is able to provide an allele translation. The array system that we are using has hundreds of different genes and thousands of different variants, not all of which have pharmacogenetic relevance, but even among those that do, there isn't necessarily an allele translation available. So these are the genes for which we have an allele translation, typically star allele nomenclature, but not exclusively. Most of these are related to drug metabolism or effect on drug metabolism. However, there are a number of other genes for which our vendor has content for. They're also very important and have pharmacogenetic implications, possibly within the area of pharmacokinetic or pharmacodynamic relevance. So we want to make sure that even if we don't have direct allele translation and we have content available for other pharmacogenetic relevant genes and variants, we want to include those as well to the extent that we can. We also look at copy number variation for CYP2D6, which commonly many laboratories do because of the relevance for that. And we also look at 7 different HLA alleles that have relevance for pharmacogenetic guidelines. So in total, we are looking at 54 different genes, hundreds of different variants that's related to more than 240 drugs in our hospital formulary. The next 2 slides are busy slides. I don't expect you to read these in any detail, but you're welcome to look at this afterwards. The point that I want to make about these 2 slides is the fact that although we're looking at many genes, it's very difficult to focus on only some and not find relevance for others, which is requiring a broader approach. So for example, for patients who are treated in our psychiatry division, many of the drugs that these patients may be on will fall into the areas of CYP2C19 or CYP2D6. However, there will also be relevance for other genes and variants outside that range. So if you're only going to focus on this, you could be missing some opportunities for those patients who are being treated with non-psychiatric medications. The same thing is true for patients who are treated with cancer. Certainly, if it's a patient who has a newly diagnosed leukemia, we want to make sure that they have information related to TPMT or NUDT15 for those thiopurine medications that those patients may be on. However, we all know that patients who are treated for cancer will have other health challenges. They may have infections and may want to be prescribed antibiotics. So therefore, it's important to look at those genes such as MTRNR1 that have relevance to the antibiotics, or also patients who may undergo surgery and therefore will be receiving anesthetics. So therefore, genes like RYR1 will become important as well. So we don't want to hyper-focus on only those drugs that the patients need for the specific therapy, but we also want to make sure that they are covered for other medications as well. And in this particular diagram, I won't go into any detail, but this is basically information that is obtained from CPIC. You can go to their website and describes the prioritization of reporting for levels of evidence for different recommendations. So they describe a very arduous process, but at the end, what we want to focus on is CPIC level A or B when we make recommendations. Level C is a little bit more tenuous because there's not as much information available that to make those recommendations, but over time this may shift to a level A or B as more information becomes involved. So I would encourage everyone to look at this particular site and this information to give you a better perspective. And with that, I will hand over the rest of the discussion to Dr. Mancuso, who will talk about the education for our providers and families.
Thank you, Dr. Melnick. When you look at any of these processes that look very complicated, in reality, it's just a complex process. Education is crucial for the success of these programs, and more when you're trying to explain pharmacogenomics or applying them into clinical care, education becomes crucial. It involves training of providers, patients, families, and you have to be able to provide them with the knowledge and resources that they need in order to understand not only pharmacogenomics, but the process that is involved in the testing and the returning of the results. Before rolling our program, we implemented extensive training for all providers through our medical education. What you see on the slide is an example of that. We provide guides on how to access our system. We also reach out to nurses and other healthcare staff involved in the test ordering. And we supplied educational materials and also conducted personalized training sessions. And we set up online resources for any follow-up questions that they may have. And also we conducted so I wanted to show you the guide for the physicians to access. And also for patients and families, we conducted, we had a website, but also we conducted other educational efforts for patients and families. And we created a brochure for pharmacogenomics and launched a website, as you see in your screen, that is hosted in our precision medicine program, and it provides all these additional resources for our patients and families to access. As I mentioned at the beginning, education is crucial, and this type of information that you need to convey is complex and also requires a multidisciplinary approach. And you should not forget that not only you are trying to educate providers, but also you need to educate the patients and the families. Now I would like to show you how our providers interact with our program. How is the physician experience, not only the patient experience. And as Dr. Melnick mentioned at the beginning of the presentation, it is important for the information to be readily available and also to be actionable for the providers. Our clinical decision support, as you see on the slide, is designed with that in mind, and it's also designed to send out alerts. That you see on the screen is one of the alerts that there are for each medication that has a different strength of evidence. And when a patient pharmacogenomic profile is aligned with a specific drug gene pair and the provider is going to order a medication, the system will fire an alert and will offer recommendations. This allows the providers to make informed decisions and adjust the prescriptions that are based on their clinical judgment. What I would like to point out is the system will encourage the proactive decision, but it doesn't mandate the changes. So it's not a hard stop for the provider. So it enables the provider to act based on the information that is presented to them. If the provider would like to further explore the genotype profile, I will show you in the next slide. They will find the detailed information on medication interactions. As you can see, there is a link that you can click for supporting evidence, and the patient genotypes are also displayed, and there are links to provide more information for the provider. Additional clinical guidelines are readily available for those providers that would like to further explore. And this comprehensive approach ensures that all the stakeholders have the necessary knowledge and tools to effectively use pharmacogenomics in patient care. So we recommend against, and we very strongly believe this, that pharmacogenomic tested into clinical practice without having a CDS system, a clinical decision support system, should not be implemented. And we are almost at the end of our presentation, and Dr. Melnick will present some closing remarks. I wanted to show you one more slide. This is how the information is presented as well for the providers, and I'll pass it on right now to Dr. Melnick.
Thank you, Dr. Mancuso. So in terms of future directions, where pharmacogenetics is going, from the laboratory end, many more individuals are looking at next-gen sequencing as an opportunity for pharmacogenetics. It has some advantages in that it's more customizable, which we are finding. We recently installed an NGS system in our laboratory. It also has a much quicker turnaround time, but it's not as comprehensive, or to make it more comprehensive is a lot more difficult. So I find that NGS and microarray can be very complementary depending on what your interests are. There's going to be an even greater role for informatics, particularly in the AI or machine learning sphere. We are learning in recent years the fact that many different genes and alleles can impact one particular medication. So in order to have a really comprehensive view, requires not only the testing for each of these, but also the managing of all the data and the interactions that occur. In order to do that, certainly AI methodologies or machine learning tools will become very important in the future to manage that level of data. And finally, the potential impact on pediatric care and personalized medicine, we believe will be realized through preemptive testing for our patients or before they become patients, children in our communities, so that when they do enter into the system, they will have the information readily available. So in conclusion, we believe that PGX continues to evolve, enhancing patient care and providing more precise and individualized treatment for our patients. Clearly, establishing a program like this requires a multidisciplinary team, and that has to be started in that fashion. The analytic stages of the program at each step must be standardized, but as we've mentioned, all of the heavy lifting has been done by the various organizations that were referred to, and this is really vital and it really needs to become a part and integrated into the program. There's no ideal solution. The program should be tailored to everyone's specific needs and the needs of the institution. But beginning the journey is best if it is most comfortable for the individual institution, and then growing it from there. It's never going to be complete when you start. It's going to be something that evolves. And this is very good advice from one of my favorite 18th century philosophers, Voltaire, who said the perfect is the enemy of the good. Meaning in our case, don't wait until you have a perfect situation to begin the process, because you'll never be able to start it. Start with something you feel comfortable with, let it grow and evolve, and you will eventually get there. Thank you very much. And also, before I get there, here are some references to make it easy for you. And I want to acknowledge many members of our different teams. Obviously, lab and pharmacy, we've talked a great deal about, but we're grateful to our IT colleagues for the support that they have provided us, all of our clinical colleagues who provided input and worked with us in the development of this process. Hospital administration has been very gracious in providing us the tools that we need to begin this journey and continue it. Our marketing team, our legal and regulatory colleagues, compliance team, finance, and patient access. So you can see that having a very robust multidisciplinary group to work with is vital in order to have a successful program. Thank you very much for your attention, and we look forward to any questions.
Thank you, gentlemen, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here asks, what is the anticipated turnaround time for PGX testing from order to result, and what factors necessitate rapid turnaround times?
Okay, I can take that. The actual runtime, as I mentioned, in the laboratory can vary, but with microarray testing can take 4 or 5 days. But the order of the test may occur the preceding week, so that extends the time. So in our case, it could be anywhere from 7 to 10 days with the level of testing that we do. Other methodologies like NGS can compress that time frame to a large extent. But in typical fashion, I would say 7 to 10 days is a reasonable turnaround time. But again, going to outside vendors, that can be extended. As far as the need, there are certain areas, for example, in psychiatry, when a patient enters into the psychiatry department for treatment, they're evaluated, obviously diagnosed. Typically, psychiatrists will provide empirical therapy based on their judgment of the patient. It may or may not be optimal, and they may not learn whether or not the therapy is optimal for some time. So in those cases, it's very important to get optimal therapy up front as close as possible to the time of diagnosis. Same thing is largely true in oncology. Patient may come in being treated for leukemia, and you want to make sure that they have the information available in a much shorter time frame before therapy can begin, or also the use of other therapies along the way during their induction or consolidation phases for treatment. So those are key areas we think are important to have more rapid turnaround.
Great, thank you. All right, our next question here asks, is in-house testing a requirement for a comprehensive PGX program?
I can take that as well. It is not a requirement. And many institutions that I'm aware of who have a pharmacogenetics program will refer their testing out to another laboratory. They will work with a vendor such as we have for informatics and will feed the information to the institution. So the way that would work is the external vendor who does the testing will work with another company, feed the information the way that we are doing, and that information will get passed on to the hospital. With that workflow, it can be a little bit more cumbersome, but it is definitely doable. The other thing is that it will be much more expensive, because when referring testing out to other vendors, and we've looked at this very early on before we made the decision to do in-house lab testing, we found that the cost for sending out the testing could be 4 or 5 times more for each patient. So that's a substantial difference in cost. So that has to be evaluated as well.
Yes. All right. Now, how is pharmacy integrated into decision support?
I can take that one.
Please.
So pharmacy is very integrated into the decision support in many ways. And we have a few pharmacies that interact with our providers that are experts on pharmacogenomics. And they collaborate by providing input on the medication alerts that we build and the dosing guidelines that we provide. They do the therapeutic monitoring that is subsequent to diagnostic from pharmacogenomics. And of course, they work with formulary management for the drugs and that we include in our panels. And they also provide education and return the tests back to the patients. So pharmacy is very integrated into our not only decision support, but in many interdisciplinary collaborations that we have with providers. So the integration of pharmacy into the decision support is crucial because, and I see this in many organizations, that the successful pharmacogenomic implementations are mostly led by pharmacists.
Thank you. And I want to thank our audience for all these great questions coming in. We do have a few more minutes here, so we'll answer a few more questions. This next question asks, what clinical disciplines are prominent users of preemptive PGX and why? Is system-wide preemptive PGX of value?
I can do one. So what we've seen in clinical practice is psychiatry, oncology, neurology. Those are some of the ones that come to my mind. Those are the bigger users, mainly because the guidelines that are available that affect the drug utilization for those specialties. And what was the second question? So it was a long question.
No problem. The second part asks, why and is the system-wide preemptive PGX of value?
Yeah, we truly believe that the preemptive implementation has to be eventually system-wide, and because that's what we discussed on the presentation, that pharmacogenomics can provide a significant value in healthcare by enhancing the efficacy of the drugs, reducing adverse drug reactions, and ultimately they contribute to personalized medicine. And they also, the information that we obtain can be also used for research purposes and leading to the discovery of new target drugs. And so there's a lot of information available when you perform this. However, the word of caution and what we try to convey in the presentation is that even though the system-wide application offers substantial benefits, there is a potential benefits. The implementation and the integration into the healthcare system requires careful planning and a lot of other considerations that we try to show you in the presentation.
Great, thank you. Now I've got another two-part question here. This question asks, what is the difference between microarray versus NGS versus RT-PCR for PGX testing? And what are the factors to consider when making a choice?
That's a question for Dr. Mel.
Okay, well, the microarray system is certainly the one we're using, and I think others as well. They are much more comprehensive. It's possible to look at literally hundreds of different genes and literally thousands of different variants. Even though we may not use all of them, that's an important opportunity, because over time there will be more content for things that we may have already tested, and then information will become available. And why that's important is that if you can imagine over the next 5 years, a patient may have already had a profile done, which will have a lot of information. Let's say there's new content available. It won't be necessary to reacquire a sample from a patient. We can take the same data file, run it through an upgraded version of the software, and obtain new information for the same patient, and then have it evaluated and have more current information provided into the EMR. So there's an opportunity to be able to do that. And also there's very good resolution with microarray techniques. There's a lot of redundancy built into the system. So the quality is usually very excellent. With NGS, there's another opportunity, and it's complementary in many ways. Unlike the microarray system, it's not that you can't customize it. So that the array comes to you from the vendor, and then basically you're using it as it's constructed, but it's so broad that you'll find what you need within it. With NGS, you have the ability, even though it can be provided by a vendor, you can customize it. So it's amenable to customization. The other point is, at least with the system that we are using, has a much more rapid turnaround time. So basically we can complete an analysis for a large number of patients in 24 hours. So it won't be as comprehensive, but it can also be very targeted. So I guess partly to the second question, depending on your interest, if you want a very broad approach and you're not pressured by time, microarray is clearly the best way to go for preemptive pharmacogenetics. But in those cases where you want to get information to clinicians, say in psychiatry or oncology or neurology or cardiology, you want to be able to get information much more quickly and a bit more focused. NGS is a very good way to do that. RT-PCR can also lead to some level of customization, but it's certainly not as comprehensive as microarray testing.
Great, thank you. All right. Now, are interpretive reports that provide details about genetic variation of drug metabolism without patient-specific gene-drug interactions sufficient?
Well, they can certainly be very valuable because you're getting information about the patient's genotype and phenotypic information. But what's lacking typically in the reports, even though many companies that provide reports will try to give you some information, is you have to look at the gene-drug interactions for that particular patient and what that will mean. That's why having a broad informatics approach, which is tailored specifically for the patient, and having the decision support for the physician immediate without having to read through all of this is of value. Now, it's fine to do that. What I'm saying is, when you get a report, and say you get a report that you generate through PharmCat, for example, even if you're doing it internally, you do your own testing, you take the VCF file, put it into PharmCat, you will get very good recommendations. But ultimately, what you can get information about a metabolizer status, but it doesn't give you the precision that you really need to match that information with the specific patient and their ultimate genotype, and then be able to make a very good drug, have good drug guidance. Yeah, so that's the difference. Maybe you want to.
No, I wanted to add that is one of the reasons that throughout the presentation we try to highlight that the need of clinical decision support. It makes things easier for the providers. The provider don't have to understand what a CYP2D6 means or what is an intermediate metabolizer. They want to know what is the action that they need to take care of that patient. So that is the translational part that is missing in many other implementations, that often is the reason these implementations fail is because they don't have a clinical decision support. So we recommend to have one of those available.
Great. Well, thank you, gentlemen. We have time for one more question here, so we'll wrap up after this one. What strategies are being implemented to educate healthcare professionals about pharmacogenomics and its clinical applications, ensuring they can effectively utilize these tests in patient care?
I went over the presentation on a few of those that we have implemented. We provide for providers, we did educational sessions. We went to their divisional meetings. Dr. Melnick and I have presented pharmacogenomics and the value of integrating pharmacogenomics into clinical care. And for patients and families, we provided a website with the frequently asked questions and a brochure that is available. Beyond that, there's incredible work that is being done at the NIH level with pharmacogenomics. Dr. Philip MP, right? MP. Dr. Philip MP is the director for that is leading this educational effort. So, and it is available online. You can go to the website, and this is a great resource for clinicians and other. It's actually for anybody that wants to understand pharmacogenomics and the implications of pharmacogenomics. It's a great resource, so I will recommend that as well. And that's pretty much how we approach the educational piece. I don't know if you have anything to add.
Great. Well, that will conclude our session for today. So I want to thank you again, Dr. Stephen Melnick and Dr. David Mancuso, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We will encourage you to share that email with your colleagues who may have missed today's live event. Upon conclusion of this webinar, a survey will pop up, and your feedback is greatly appreciated. To all our friends out there, thank you for joining us. Have a great day, everyone. Goodbye.
Thank you.
Thank you.